Okay, I want everyone listening to do something for me. Just for a second, unless you're driving. Please don't. Picture that diagram from your old biology textbook. You know the one I'm talking about, the tongue map.
Oh, the classic, the colorful little chart.
Right, sweet on the tip, salty on the sides, bitter way in the back. And they taught it to us like it was, scientific gospel, like it was undeniable.
It was presented as fact.
Well, I am here to tell you that believing in that map is basically the culinary equivalent of believing the Earth is flat. Or, I don't know, that Star Wars episode is the best movie in the franchise.
It is a total myth, a complete fabrication that has just somehow survived for decades.
Which is why today we are tearing up that old map. We are jumping into the deep dive lab to decode this massive stack of research simply titled Exhaustive Literature Survey on the Sense of Taste.
And when they say exhaustive, they really mean it.
They do, and this isn't just about why you enjoy a good pizza. This is about quantum-level molecular locks, phantom signals, and receptors hiding in places you would never, ever expect.
That's the thing that really stands out. We tend to think of taste as this simple pleasure thing, but when you actually look at the data, the specific proteins and signaling pathways, it's one of the most sophisticated chemical scanning systems in all of biology.
I was reading through this section on cellular mechanics, and it honestly reminded me of the control panel on the Millennium Falcon.
Huh.
Oh, no, seriously. To the untrained eye, it just looks like this chaotic mess of wires and blinking buttons, but every single one of those switches has a specific, probably life-saving purpose.
That is a surprisingly accurate analogy. It's a system that's built for survival first and enjoyment second.
So let's pop the hood. Let's talk about the hardware. Because for years, I just thought taste was, well, the tongue. But the authors here point out that's a massive oversimplification.
It is. The actual sensors, what we call taste buds, they aren't just on the tongue. The literature confirms we have these functional taste receptors in the soft palate, the throat, the epiglottis,
The epiglottis.
and even down into the upper part of the esophagus.
So we are chemically analyzing our food like halfway down the hatch.
Precisely. This extra-oral distribution suggests that taste is doing a lot more than just giving you that initial yum or yuck. It's monitoring the food as it moves through the entire system.
Okay, so let's zoom in on the taste bud itself. Because the diagram is always just showed as a little bump, but that's not quite right, is it?
No, the bump you see is the papilla. The taste bud is actually buried inside that tissue, and it's shaped a bit like an onion bulb. At the very top, there's a tiny opening called the taste pore.
So that's like the airlock.
That's a great way to visualize it. The chemical visitors, or tastants, they have to pass through that little fluid-filled funnel to get clearance to meet the crew inside.
And inside that little bulb, you've got a whole team, like 50 to 150 specialized cells packed in there.
And this is where that Millennium Falcon complexity really kicks in. The survey breaks these cells down into specific types I through IV, and they aren't just copies of each other. They have jobs.
A higher...
Absolutely.
Yeah.
So let's look at type I cells. The authors describe them as the support crew. They're a lot like glial cells in the brain. Their main job is to be the janitors.
The janitors.
Yeah, they clean up neurotransmitters, they keep the ionic environment stable so the other, more specialized cells can do their work.
Okay, important, but not the rock stars. The heavy lifting seems to be done by types 2 and 3.
Correct. Type 2 cells are the specialists. They handle the big three: sweet, bitter, and umami. But here is the fascinating part that the researchers highlight. Type 2 cells don't have normal wiring.
I had to reread that part twice. If they don't have a synaptic connection, how do they even talk to the brain?
They broadcast. When a type 2 cell detects, say, sugar, it releases a chemical signal, specifically ATP, into the area around it. It's a paracrine signal.
So instead of plugging in an Ethernet cable, they're basically blasting a Wi-Fi signal to the neighborhood.
Exactly. They spray the signal. Now compare that to type 3 cells, the electricians. They handle sour. And these cells do form actual synaptic junctions with the nerve terminals. They plug right in.
So sour gets a hard line, but sweet is on Wi-Fi. Is there a reason for that?
It allows for modulation. That ATP signal from the type 2 cells can actually influence the type 3 cells nearby. It creates this local processing network before the signal even leaves the tongue.
That is wild. And then you've got type 4 sort of lurking in the background.
The clones.
I knew it.
Type 4 are the basal stem cells. You have to remember, the inside of your mouth is a war zone. You've got burns from hot coffee, friction from chewing, all sorts of chemicals. Taste cells die incredibly fast.
So type 4's job is just to replace the fallen soldiers.
Constantly. They divide and differentiate to replace the entire taste bud every 1 to 2 weeks.
So it's like Doctor Who. The taste bud regenerates, it has the same job, but poof, it's wearing a completely new face.
It's a relentless cycle. You
Yeah.
literally have a new sense of taste every fortnight.
Okay, so we have the crew. Now let's talk about what they're detecting. The infinity stones of flavor. We all know the classic five. Sweet, salty, sour, bitter, umami. But this survey spends a lot of time on a sixth one.
Yes. The evidence for fat taste is becoming, well, overwhelming,
They call it Ole Augustus.
which sounds like a Roman emperor, doesn't it?
All hail Emperor Ole Augustus. But seriously, I always just thought fat was a texture thing. You know, that creamy mouthfeel.
And that's part of it. That's the somatosensory part, the touch. But Ole Augustus is a dedicated chemical detection system for fatty acids.
And the authors back this up with some pretty specific genetic evidence.
They do. They highlight a receptor called CD36. It's a fatty acid transporter. And the studies show if you delete the gene for CD36 in mice, they completely lose their preference for fat.
So the mouse just doesn't care about the butter anymore.
Exactly. It just treats it like anything else. There's another gene, too, called STIM1 that controls calcium signals. You knock that out, the preference for fat just disappears.
Which proves the body is chemically scanning for high-energy fuel, completely separate from the texture.
Precisely. It's a survival algorithm.
Okay, let's unpack the mechanics of this, the lock and key. Because the descriptions in this survey are just incredible. Let's start with the good stuff. Sweet and umami.
Right. These rely on the T1R family of receptors. They work in pairs, called heterodimers. Imagine two proteins that are sort of hugging each other.
A protein hug. I like it.
So if you have T1R2 and T1R3 hugging, that detects sweet. If you swap out the first one for T1R1, see T1R1 and T1R3, that detects umami.
But visually, these things aren't just passive holes.
No, they are aggressive. They have what the authors describe as a Venus flytrap domain.
A Venus flytrap domain.
Yes. The molecular structure literally looks like an open jaw. When a sugar molecule or an amino acid floats in, those jaws snap shut around it.
That is so cool. It's not a sensor, it's a molecular trap.
It's very dynamic. Now compare that to bitter. That's the T2R family.
The defense system.
Right. Humans have about 25 different types of bitter receptors. And unlike sweet, where one receptor type catches pretty much all sugars, bitter receptors are tuned to all these different specific threats.
Because in nature, bitter usually means poison.
Exactly. We need to detect a huge variety of bacterial metabolites, plant alkaloids, toxins.
That's like the shields on the Enterprise from Star Trek. You can't just block lasers. You gotta block photon torpedoes, tractor beams, anomalies. The system has to be versatile to keep the ship safe.
That's a fair comparison. And the key difference is sensitivity. You need a lot of sugar to trigger the sweet receptor, but you only need a tiny, tiny amount of a toxin to trigger the bitter one.
Speaking of sensitivity, the mechanism for salty was one of the most elegant things in the report. It's a dual system.
It uses the Goldilocks principle. We have a low salt sensor called ENS. When salt is low, this channel opens, and the signal to the brain is good. It's appetitive.
That's me with potato chips. Just
Yeah.
one more.
Right. But if the salt concentration gets too high, I think the survey mentions over 150 millimolar, a completely different pathway kicks in, and it becomes aversive.
It stops tasting good and starts tasting, like, painful.
Correct. The body automatically stops you from overdosing on salt without you having to think about it.
Yeah.
It's a hardware limit built right into the tongue.
But the one that really, truly blew my mind in this report was sour. There is a plot twist with sour.
The off response.
Explain this, because it sounds completely backward.
So the candidate receptor is a channel called PKD1L3, PKD2L1. The research indicates that these channels often don't open when the acid first hits them.
Okay.
They open when the acid is removed,
Wait, hold on. So I bite into a lemon, and the receptor waits until I swallowed a fire.
or until your saliva washes the acid away and the pH goes back up. That is when the channel fires.
That is the horror movie villain who jumps out of the closet just when you think you're safe. Or like Wile E. Coyote, who runs off a cliff and doesn't fall until he looks down.
It's a delayed reaction mechanism. The thinking is that this might help the body distinguish between different types of acids, or maybe it signals that the immediate threat is clearing.
Nature is so weird. Okay, speaking of nature being weird, let's talk about the mutants among us.
I assume you mean this section on genetic variability.
I do. The report goes deep into the genetics of why some people hate broccoli and others can't get enough of it.
It focuses on a specific gene called TAS2R38. It's a bitter receptor gene. The survey details two main versions or haplotypes. You have the PAV version and the AVI version.
PAV and AVI sounds like old video file formats.
If you have the PAV version, you're a taster. You perceive a specific chemical called PTC as intensely bitter. If you have the AVI version, you're a non-taster. To you, it literally just tastes like water.
And then there are the supertasters.
Right. These individuals often have that PIV gene, but they also have a higher density of fungiform papillae, so more taste buds per square inch.
So it's like having spidey sense. Everything is just dialed up to 11.
Exactly. Coffee is way too bitter. Grapefruit is aggressive. Even certain textures can feel overwhelming.
Sounds exhausting, honestly. I'm happy being a medium taster. But talking about genetics, we have to talk about cats.
The Felinidae family.
Yes. This is my favorite fun fact. Cats can't taste sweet.
No, they can't. The literature notes they have a mutation in that T1R2 gene we talked about. For them, it's a pseudogene. It's broken.
So while my dog will go crazy for a donut if I drop it, my cat just looks at it like it's a rock.
Because to a cat, sugar has no signal. They're obligate carnivores. Evolutionarily, they didn't need to detect carbs. They needed to detect protein and potential toxins in meat.
So they're the Voldemorts of the animal kingdom, unable to feel joy sweetness, only focused on the dark arts of protein and bitter, rancid meat.
Well, they actually have an enhanced bitter sense compared to us. They can taste things in meat-specific amino acids linked to spoilage that we would completely miss.
Okay, remind me never to share steak with my cat. Too critical.
Now, this is where the survey pivots to something really profound. We've been talking about the tongue, the receptors, but the flavor of food, that isn't happening in your mouth. That's happening in your brain.
The matrix of flavor. This is that difference between taste and flavor.
Correct. Taste is just those 5 or 6 modalities we discussed. Flavor is the brain integrating taste, touch, temperature, and most importantly, smell.
But not just smelling with your nose, like sniffing a flower.
Right. There's orthonasal olfaction, sniffing through your nostrils, and then there's retronasal olfaction. When you chew, volatile molecules float up the back of your throat into your nasal cavity.
So when I say, this candy tastes like strawberry, my tongue is just getting sweet and sour. It's my nose working from the back door that's saying strawberry.
Exactly.
It's a ventriloquist act. The nose is doing all the work, but the brain throws the voice to the mouth so we think the flavor is coming from the tongue.
That is a perfect way to describe it. It's a sensory illusion. And the brain regions involved are fascinating. The authors identify the ventral pallidum as the hedonic hotspot.
I love that name, the hedonic hotspot.
It's a tiny area that lights up specifically for pleasure.
The one ring of the brain, the center of desire.
When you eat something truly delicious, that's where the liking happens. And interestingly, temperature plays a huge role in how these signals get there. We call it thermal gating.
This was the life hack section of the report.
The TRPM5 channel, that universal signal booster for sweet, bitter, and umami, is heat sensitive. It opens more easily when it's warm.
Which explains why warm soda tastes so much sweeter and kind of grosser than cold soda.
Yes, and conversely, if you cool the mouth down, you can reset the threshold. The report mentions that sucking on ice cubes can actually help patients with dysgeusia distorted taste because the cold essentially reboots the channel's sensitivity.
So if your taste is acting up, try an ice cube. Who knew?
It's simple thermodynamics applied to biology.
All right, we've done the tongue, the genetics, the brain. But the report had one final twist that I genuinely did not see coming. The second brain.
The gut.
We established that taste receptors aren't just in the mouth, but the survey confirms they are deep in the stomach and the intestines. What are they doing down there? Tasting my lunch a second time.
In a way, yes. But they aren't sending flavor signals to your conscious brain. You don't taste a burrito in your stomach.
Yeah.
They're regulating your metabolism.
Give me an example.
Okay, take the bitter receptors in the gut. When they detect strong bitter compounds, which, signal toxins, they trigger a response to slow down digestion.
They stop the conveyor belt.
Exactly. They're saying, hold on, let's analyze this, don't absorb it yet. On the flip side, when the sweet and umami receptors in the gut detect sugar or protein, they trigger the release of hormones like GLP-1 and CCK.
GLP-1. Wait, isn't that the stuff in the new weight loss drugs everyone's talking about, like Ozempic?
The very same. Those drugs mimic the hormone that your gut naturally releases when it tastes nutrients. That hormone is what tells your brain we are full, stop eating.
So my stomach is tasting my food to calculate the calorie count and tell my brain when to put the fork down.
It's a metabolic feedback loop.
Yeah.
And it happens completely without your conscious permission.
That's like the force. It's this invisible energy field binding the whole body together. The mouth talks to the brain talks to the gut talks right back to the brain.
It's a unified system. We often think of taste as just a luxury, something for a Michelin star meal. But this research just shows it's a survival mechanism, a toxin detector, and a nutrient calculator all rolled into one.
So let's bring this deep dive in for a landing. When you look at this whole exhaustive survey, what are the big takeaways for you?
Three things really stand out to me. First, just the sheer complexity. We aren't dealing with a simple tongue map. We're dealing with quantum-like locking mechanisms, these Venus flytrap receptors, and 6 distinct modalities, including fat.
Okay. And second,
The illusion. Flavor is a construct. It's a simulation your brain creates by merging smell, touch, and taste. We don't experience reality directly. We experience the brain's story of it.
and the third.
The scope. We taste with our whole body. From the throat all the way down to the gut, our biology is constantly scanning the chemical world to keep us alive.
It really makes you think. If your stomach can taste things without you knowing, and your brain is basically hallucinating strawberry flavor based on smell, how much of your favorite meal is even real? And how much is just a neurological story your brain tells you?
That is the ultimate question.
Something to chew on during your next meal. Thank you for entering the Deep Dive Lab with us today.
It's been a pleasure.
If you enjoyed having your mind blown, go ahead and hit that subscribe button. We've got plenty more mysteries to unpack. Until next time.
Stay curious.